Okay, so just picking up where we left off the other day. Last stuff that we talked about were the innate immune defense mechanisms that we have. Remember, innate means that we were born with it. And the other term that we can describe that form of immunity is non-specific, because it involves essentially just fighting anything the same exact way, no matter what. If it's a virus, a bacteria, a fungus, we're going to respond to it the same way. It involves things like keeping stuff out using our skin, cilia in our throat, second defense mechanisms or second lines of defense, which include like interferons and various cells. And then the last stuff that we talked about was inflammation and fever, which kind of go hand in hand. The various components of the innate defense mechanisms were listed here on table 22.1 from the last or that we covered last time. We're ready now to talk about adaptive immunity, which is quite a bit more complicated. It refers to our body's ability to defend itself against specific invading agents such as bacteria, toxins, virus, foreign tissues, substances that are recognized as foreign and provoke immune responses are called antigens. And there's that word that we've seen here the last couple class periods. Two properties distinguish adaptive immunity from innate, and we talked about this the other day, and I was just kind of mentioning it. But to reiterate, specificity, which means that we are specifically going after something, and we would call it an antigen, a foreign molecule. This also involves distinguishing self from non-self molecules. So, the word specific begins that word specificity. So that's kind of what you can think of. We're looking for something specific, a specific type of virus, a specific type of virus, whatever, or bacteria, whatever. Okay. And then memory, being able to remember something we've previously encountered, so that second encounter prompts an even more rapid and vigorous response. We'll get into kind of just how much more rapid and whatnot that all is here in a little bit. Okay. Adaptive immunity involves the major lymphocytes B and T cells that we've mentioned a variety of times. B lymphocytes and T lymphocytes develop immunocompetence, which I talked a little bit about the other day, the ability to carry out adaptive immune responses. We learned that B and T cells undergo this or develop immunocompetence in the primary lymphatic organs, which are the red bone marrow and your thymus. Okay, and I compared that to like a soldier going off to basic training and getting skilled as a competent soldier. In the case of these B's and T cells, what they do to become immunocompetent is create or make several distinctive proteins that they insert into their plasma membrane. Some of these proteins function as antigen receptors, which are molecules that are able to bind to the specific antigens, able to recognize and bind to them or interact with them. An important process involved in immunocompetence is negative selection, which is actually where our body destroys both the T and B cells that would be prone to attack against its own antigens, our own host antigens. So in other words, in doing so, that prevents the T cells, B cells from, damaging our own body and killing our own cells, which unfortunately does happen from time to time, which we'll talk a little bit about towards the end of the lecture. So let's talk a little bit more about the different lymphocytes. We've got the T's, we've got the B's, okay? Both of which, just like any other blood cell, originate in the red bone marrow, whereas the B cells stay in the bone marrow and develop there. The T cells mature in the thymus, so they're derived from the thymus, so it's kind of easy to remember T for thymus. And they produce what we call cell-mediated immunity. Okay, they're way more prevalent. About 70 to 80% of the circulating lymphocytes in our blood are T cells. They also can become associated with the structures that we learned about the other day, like the lymph nodes, the spleen, and other lymphoid tissues, like your appendix and your tonsils and whatnot. Okay, and before leaving the thymus, those T cells develop immunocompetence, and typically they mature and exit the thymus as one of the following two types of cells: helper T's, and what I usually call them are killer T's, which also go by cytotoxic, which is kind of a fancy word for killer. Okay, They also go by the terms CD4 cells and CD8 cells, and the CD4, CD8 stuff just refers to the type of proteins are in their plasma membranes, so the type of receptors that they have. The helper Ts, they help. Pretty common sense as to why I would call them that then. They help activate other T lymphocytes, or they can help activate B lymphocytes to become plasma cells. We'll talk a little bit more about those at the bottom of the page, as you can see. Those killer T cells combine with the antigen on the surface of the foreign cell, causing lysis of the foreign cell and the release of cytokines, which are chemical messengers of the immune system. So they help kill cells, and they can release the cytokines that help with messaging. There's also two other types of T cells, memory Ts and regulatory Ts. Memory Ts remain inactive until future exposure to the same antigen occurs. And if they do encounter that same antigen at a later time, they're able to respond very swiftly because there are thousands of them in our body. Okay. And then regulatory T cells are those that help prevent autoimmune diseases, where essentially the immune system attacks itself. And so regulatory T cells provide a break, and you can see the quotes there around that word break, on our specific immune response by inhibiting killer T and B cells. So they kind of keep the soldiers in check and prevent them from attacking our normal body antigens. Unfortunately, as I mentioned, sometimes that does happen where the immune system attacks itself, and so those regulatory T cells fail to kind of complete their job. All right, so that's T cells. B cells are produced in the bone marrow. They develop in the bone marrow as well. So it's easy, B for bone, whereas T was for thymus. They produce antibody-mediated immunity. They account for 20 to 30% of the circulating lymphocytes in our blood, and also become associated with our lymph nodes, spleen, and other lymphoid tissues. When they encounter a foreign antigen, it internalizes and digests it, presents the fragments of the antigen to a helper T cell, which then secretes chemical help factors that stimulate the cell to divide into two types of cells. Most of those daughter cells that are produced are known as plasma cells. Okay, and the very important factors or very important process that a plasma cell does is it produces the antibodies. So plasma cells are the actual producers of antibodies, which that's why B cells are involved with antibody-mediated immunity. B cells form plasma cells, which then form the antibodies. The other cells that are formed from those original B cells are called memory B cells, which can live for months to years and respond very quickly if they ever encounter the same antigen again. So they're kind of like what we saw with the memory T cells. Let's talk a little bit more about cell-mediated immunity, and then we'll talk a little bit more about antibody-mediated immunity. Cell-mediated immunity is particularly effective against intracellular pathogens, which means that the pathogen is on the inside of the cell. So like a virus has infected a cell or invaded a cell. Okay. They also are effective against cancer cells and foreign tissue transplants. Okay, in the case of cell-mediated immunity, it begins with the activation of a small number of T cells by a specific antigen. Clone cells are then formed, which then recognize the same antigen as the original cell. Some of these clones become effector cells, while others become memory cells. Effector cells carry out the immune responses that ultimately result in the elimination of the intruder. Effector cells include those active helper Ts that I just mentioned, which are part of a helper T cell clone, and then the active cytotoxic or killer T cells, which are part of a cytotoxic T cell clone. Memory cells are going to reside in lymphatic tissues, ready to respond more rapidly and forcefully if the same antigen invades the body again. The thousands of memory cells will respond to the antigen by proliferating and differentiating into more effector and more memory cells. These include the memory helper Ts, which are part of a helper T cell clone, memory cytotoxic T cells, which are part of a cytotoxic T cell clone, and memory B cells, which are part of the B cell clone. These cells typically have long lives, and we're talking decades. So an extremely long time, especially when you think about how long cells typically live. When it comes to cell-mediated immunity, it involves those killer or cytotoxic T cells. They battle foreign invaders in cell-mediated immune responses. They are going to target infected target cells, cancer cells, and transplanted cells. They recognize and attach these target cells and then deliver a lethal hit that kills the target cells. Okay. They can kill infected target body cells in a similar way as a natural killer cell does, which we talked about that process the other day. Major difference is that cytotoxic T cells have specific receptors specific for a particular microbe and thus kill only target body cells infected with one particular type of microbe, whereas natural killer cells attack a wide variety of microbe infected body cells. There are two principal mechanisms that those killer T cells utilize to kill infected target cells. Okay, using the receptors on the surfaces, those killer T's or cytotoxic T cells recognize and bind to infected target cells that have microbial antigens displayed on their surface. The cytotoxic T cell then releases those same different things we talked about the other day, grainzymes, which are protein digesting enzymes that trigger apoptosis, okay, which we talked about the other day when we learned about the functions of natural killer cells. Once the infected cells are destroyed, the released microbes are killed by phagocytes. So it's basically the same thing as what we learned the other day. Okay. Alternatively, cytotoxic T cells also can bind to infected body cells and release two proteins from their granules, perforin and granulysin. We talked about perforin the other day, but not granulysin. Perforin is going to insert itself into the plasma membrane of the target cell and create channels or like perforations or holes in them. As a result, extracellular fluid flows into it and cytolysis occurs. So in other words, like the balloon filling up with too much water or air and it pops, explodes. Okay. Other granules in those cytotoxic T cells release granulysin, enters the channels and destroys the microbes by creating holes in their plasma membrane. Cytotoxic T cells may also destroy target cells by releasing a toxic molecule called lymphotoxin, which activates enzymes in the target cell. These enzymes cause the target cells to DNA to fragment and the cell then dies. If it's got no DNA, it's going to not survive. In addition, cytotoxic T cells secrete gamma interferon, which attracts and activates phagocytic cells. There's also macrophage migration inhibition factor, which prevents migration of phagocytes from the infection area. And then after detaching from a target cell, a cytotoxic T cell can just start the whole process over by seeking out and destroying another target cell. So it's pretty cool how that all works, all the different chemicals involved there. When it comes to antibody-mediated immunity, it's mainly going to be utilizing stuff against extracellular pathogens. So we said cell-mediated was intracellular pathogens. In the case of antibody-mediated, it's extracellular pathogens, which means that they are pathogens on the outside of cells. Okay, so like virus, bacteria, fungi that are in our body fluids, but not inside the cells themselves. Once again, it involves B cells becoming plasma cells or plasmacytes, as you'll sometimes see them listed, which then make the antibodies. We haven't talked too much about antibodies, but we have mentioned them a time or two. We will talk about them more here in just a second, but they are proteins circulating in our blood and other body fluids. Each B cell produces only one specific type of antibody protein, which means that is an antibody that can bind to only one specific antigen. Okay. When an antigen binds to its antibody receptor on the B cell surface, it stimulates two things: mitotic division of the B cells, and then the specialization of the newly formed B cells into either plasma cells or the memory B cells. Once again, plasma cells are the factories for producing antibody proteins. A few days after exposure to an antigen, a plasma cell can secrete hundreds of millions of antibodies each day. That takes about 4 to 5 days until it dies off. Memory B cells do not actively participate in the initial immune response to that antigen, but they can quickly proliferate or divide and differentiate into more plasma cells and more memory B cells should the same antigen reappear at a future time. Another term for antibody-mediated immunity is humoral immunity, and this is because what we're doing in this case is typically out in the body humors, which is kind of a traditional term. Instead of calling them body humors, typically we call them body fluids now. Okay, but that's where that word humoral immunity comes from, or that's where that term comes from. Now, keep in mind, antibody-mediated immunity, cell-mediated immunity. These two processes aren't exclusively, performed separately from one another. They are processes that we're doing together. In fact, helper T cells are going to help with both types. And we can see this cooperation between the two different forms of adaptive immunity here on the next page. In most cases, when a particular antigen initially enters the body, there's only a small group of lymphocytes, which include helper Ts, killer Ts, and B cells, with the correct antigen receptors to respond to it. Okay. Depending on its location, a given antigen can provoke both types of adaptive immune responses, because in most cases, that specific antigen invading the body is not just inside cells or between cells in the fluids, it's probably getting in both places. Okay, so some copies of the antigen may be present inside body cells, which would provoke a cell-mediated immune response, whereas other copies are present in the extracellular fluid, which provokes an antibody-mediated immune response. This illustrates that cell-mediated and antibody-mediated immune responses often work together to eliminate the large number of copies of a particular antigen in the body. So looking here, we've got on the right our red bone marrow, which is, once again, where all blood cells are made, including the pre-T cells, the pre-T lymphocytes, which then go to the thymus and become mature T cells, whereas the B cells start and then finish their maturation in the bone marrow. Okay, and then as we just talked, T cells are involved with cell-mediated immunity, whereas the B cells are involved with antibody-mediated immunity. We can see the two main types of T cells here, the killer Ts and the helper Ts. Here's the B cells, which can become plasma cells that produce the antibodies, which are these little red things that look like the letter Y. Some become the B cells, the memory B cells. Okay, over here, we learned that the helper Ts not only help with the cell-mediated immunity, they also help with antibody-mediated immunity, and we can see that right in here in this portion of the figure. It gets pretty complicated when you get into all the receptors and all that wonderful stuff that's going on. Here we can see like the CD8 protein, and that's why that's another way to describe killer T cells is CD8 cells, and CD4 proteins have this, or CD4 proteins are in the CD4 helper T cells. So I won't really get into too much detail, but I just wanted to kind of point that out, and you can see it there. Let's talk a little bit more about antibodies. Once again, antibodies are in essence produced by B lymphocytes, not directly, because it's actually the plasma cells that do the production of them. We classify proteins in the gamma globulin class, which we talked about in our last lecture. They consist of 2 short or light chains and 2 long or heavy chains, as well as antigen binding sites that are highly specific for only one particular antigen. Okay, if we go to the next couple pages, we can see kind of they have some pretty cool shapes. Some of them have this nice traditional kind of Y shape to them. Some of them, to me, always looks like this is a snowflake, but there are specific sites, and they're not labeled, and that's okay, where they would bind to an antigen, and then the light heavy chains are in there as well. Okay. Now, when it binds to the antigen, its specific antibody produces triggers production. That's what I should say, not produces production. That doesn't make no sense, but it triggers production of more antibodies specific to that antigen. Okay. There are 5 classes of antibodies, each of which has a unique role. Okay. They all begin with Ig, which is short for immunoglobulin, which is just another term for antibody. And then the class is either M, A, D, G, or E. IgMs are the first antibody class to be secreted by plasma cells after an initial exposure to any antigen. IgMs are going to help activate the complement system, which, remember, is that protein collection that works like a domino effect where one knocks down the next. And this is going to trigger agglutination, which is something we talked a lot about in our last lecture, and lysis, or breakdown of the microbes. Okay. IgMs also serve as antigen receptors, and we'll kind of talk a little bit more about what agglutination and whatnot means here in just a second. IgAs we find in mucus, so places like our nose, in our saliva, lacrimal, bronchial, intestinal, and vaginal secretions. So your eyes, your throat, intestines, and then in the vagina. And these are going to help block the entrance of antigens into the body. IgDs are important in activation of B cells. IgGs are the most abundant class, protecting against bacteria and viruses by enhancing phagocytosis, neutralizing bacterial and other toxins, triggering the complement system. I would put a big fat star, highlight. It is the only class of antibody to cross the placenta from mom to fetus. So this is a way that mom can deliver some antibodies to the baby even before the baby's been subject to the outside world, and they haven't been exposed to viruses and toxins, bacteria, that kind of stuff in a natural way. So that's kind of really cool. Okay. IgEs are involved with allergies and aid in immunity against parasitic worms and other parasites. So we'll talk a little bit about them and how they trigger allergies in a little bit. So what do antibodies do, and how do they basically provide us with protection? Okay. Well, the action of the 5 classes of antibodies differ somewhat, but all of them act to disable antigens in some way. Okay. There are kind of 5 major actions of antibodies. First one is to just neutralize the antigen. The reaction of an antibody with the antigen blocks or neutralizes some bacterial toxins and prevents attachment of some viruses to body cells. So in doing so, the virus can't invade the body cell and then infect it. Okay. Agglutinating antigens. We talked about agglutination the other day with regards to red blood cells when we were talking about blood typing. Okay. Kind of the same situation occurs here. Because antibodies have 2 or more sites for binding to an antigen, the antigen antibody reaction can cross-link pathogens to one another, causing them to clump together or agglutinate. Okay. Phagocytic cells ingest agglutinated microbes more easily. And we can see that really nicely here in figure B on the next page. Each of these little brown rod-shaped structures might be a bacterial cell. Okay. And the kind of white, the blue, yellow shaded structure here is an antibody. You can see they pull them together, and so all those microbes get essentially clumped together, which then gives those macrophages, those Pac-Man cells, an easier opportunity to digest them, perform phagocytosis. Okay. Over on the right or on the left side of the page there, neutralizing antigens, what we talked about right here. All right, precipitating antigens. Antibodies can cross-link soluble antigens into complexes that are too large to stay in solution. So as a result, the complex precipitates out of the solution, making it easier for the antigen to be phagocytized. So in essence, it pulls it out of the fluid, and that way the macrophages and the neutrophils can come gobble it up. And then the letter D figure shows what happens with the complement system and how it works. Okay. Antigen antibody complexes can initiate the classical pathway of the complement system, which is one of the ways that you can activate the complement system. I won't really get into how all that works, but essentially it just is like knocking down the first domino, which then knocks down the rest of the dominoes, and that's the kind of analogy I had for the complement system. And then opsonization is where antibodies can function as opsonins that coat the surface of the microbe, which makes it easier for phagocytes to engulf and destroy it. And that's what we show here at the bottom of the page, where the microbe has become surrounded by all those antibodies, and now a phagocyte can go and engulf it and digest it much more easily. So essentially, in many cases, the antibody really doesn't do anything to kill the bacteria, the virus, or whatever itself, but it makes it easier for something else to do it. And in a lot of cases, it's a phagocyte is more likely to be able to come and gobble it up and destroy it in that manner. Okay. As you can see, table 22.3 just lists off those 5 classes, gives you a nice view of their structure and kind of describes their characteristics and functions, which we've already kind of talked about. So I'll jump ahead to talking more about some other stuff related to immunity. All right, let's talk about primary and secondary response. Okay. When a person is exposed to a particular pathogen for the first time, there is a latent period of about 5 to 10 days, so about a week before measurable amounts of specific antibodies appear in their blood. Okay, so obviously we are dealing right now still with the pandemic, and the big virus everybody wants to talk about is the coronavirus or COVID. Okay. When you are first exposed for the very first time, you've never been exposed to that virus, okay, it takes about 5 to 10 days before our body really starts pumping out antibodies to protect you against that. Well, that's a pretty significant amount of time, and it's not a quick enough response in many cases to prevent a person from getting sick, from getting the disease. And we call this sluggish response the primary response. Okay. But if you are exposed a second or third or fourth time, okay, what we're going to see is that our production of antibodies is very, very rapid. I mean, we're talking maximum antibody concentration in the blood being reached in less than 2 hours. It took 5 to 10 days, over a week in some cases, almost 2 weeks with a primary response, and now it's a couple hours. Okay. And this secondary response typically is going to be enough to prevent one from acquiring the disease. And this is where that immunological memory comes from and why it provides the basis for acquiring immunity against pathogens. One way that people can develop immunity can be described, or 4 different ways, I should say, that a person can develop immunity are listed here on the next page. There's kind of 2 different categories that we're going to talk about. How we acquire it either actively or passively, and then how we acquire it, which can either be naturally or artificially. Okay. Let's talk about active versus passive acquiring. Okay. If you acquire your immunity actively, it's because you're producing your own antibodies against that specific antigen. Okay. So we're developing our own antibodies and our own immune system cells. Whereas if it's acquired passively, it's where the antibodies are received from another person or another animal. And because there's no memory component involved, immunity that's passively acquired is only temporary, lasts until the antibodies degrade. So in the case of those IgGs, where the antibodies can cross from placenta from mom to fetus, is that going to be acquired actively or passively? Is the baby producing his own antibodies or her own antibodies, or are they getting it from someone else? Obviously from someone else, so that makes that passive. Does that make sense? Okay. Whereas if, you are exposed to the virus or whatever it is that you're going to produce the antibodies for, and you produce your own, that would be actively acquired immunity. Okay. Once again, we can also describe adaptive immunity as being acquired naturally or artificially. If it's acquired naturally, it's where they develop their immunity as a result of a natural event, such as exposure to an antigen by chance. If it's acquired artificially, the person develops immunity as a result of artificial means, which includes deliberate exposure to an antigen through vaccination. So based on these factors, there are 4 ways to acquire adaptive immunity, and they are listed here on the next page in table, whatever that is, 17.4, I think is what it is. Not that the number matters. I do actually want you to know this table though. So put a star on it, put a highlight on it, whatever you need to do. I do want you to know this for the exam next week. We have what we call active natural, active artificial, passive natural, passive artificial. Okay. Active natural immunity involves natural exposure to a microbe, okay, and then antigen recognition by B and T cells, and co-stimulation leading to the formation of antibody secreting plasma cells, cytotoxic T cells, and B and T cells. So in this case, they're producing their own immune system stuff, antibodies and B and T cells, okay, and it's natural exposure. So that's why it's active and natural. Whereas active artificial, this is like what happens when you get a vaccination. Okay, the artificial is the vaccination, and then the active means that they produce their own antibodies and immune system cells. Okay. Now, in most cases, when somebody gets a vaccination, the antigens involved there are immunogenic, but not pathogenic. So they can trigger immune responses, but they're not pathogenic in that they can't cause disease. So a lot of times it's like an attenuated virus, so it's like almost like dead, or it's weakened, so you're more likely not to actually get sick from it. Okay. Passive natural is like in the case of the transfer of IgG antibodies from mom to fetus across the placenta. So the baby's not producing its own, but those antibodies were produced naturally by mom. Same thing can actually happen with IgA antibodies when a mother breastfeeds her baby, which is really cool. And another great example of the benefits of breastfeeding, which I don't get into in great detail. But aside from being free, which formula is not, mom can deliver some of her antibodies to baby. And then passive artificial is where like an intravenous injection of antibodies is obtained from another human or an animal donor, which provides protection immediately against that antigen, but they didn't produce their own and it was artificially given to them. So that makes it passive artificial. Now you might be wondering, what about, you know, the vaccine that everybody wants to talk about now, the hot button topic of whether you should or should not get the vaccine against COVID, whether it's Moderna or any of the other types. Okay. I'm going to give you kind of a quick rundown on how that all works. I do think it's important that I do mention this. It really is very different than what you see here with regards to these and how most vaccines really traditionally have been. Like I said, in the case of most vaccines, it's been like a weakened or dead virus, whether it's like when you get a flu shot or like a chickenpox shot, which they have now. When I was a kid, you did not have those. It was actually, you had to get the chickenpox, which I had as a child. But in the case of COVID, it's a new concept. And I'll kind of break it down for you like this. So hang on one second. Instead of actually exposing the individual to the virus, they're going to do something very different. Okay. In this vaccine, it's all centered around gene expression, really. Okay. If you don't remember, gene expression is simply the process where we go from DNA to RNA to proteins, right? We've talked about this several different times this semester. Okay. Well, in the case of Moderna and the other vaccines, okay, what they can describe those as are mRNA vaccines, okay, which means that basically they are giving you a little bit of RNA in a natural way, okay, and by natural I mean in an injection, okay, which is not the virus, nor is it even the protein that the virus has on its cell surface, okay, or not cell because viruses aren't cell, but on its outer membrane, okay. And giving us this RNA, it triggers that process where we go from RNA to proteins, okay. So you're not going to get sick because you're not being injected with viruses. You're essentially going to naturally make the proteins that that virus will have. That way, if you are exposed to the virus, you've already seen those proteins and you're now already able to target them. You've already produced the correct cells, the correct antibodies, so that you can have that quicker response and are less likely to get sick. Does that make sense? Now I could get into more detail about like what protein it is that you end up making. I could talk about a lot of things, but I just wanted to kind of simplify and kind of explain it for you really quickly. Let's talk a little about some bad parts of our immune system. And some unfortunate conditions, what we call autoimmune diseases, which are diseases that occur when our immune system fails to display what we call self-tolerance. And so those natural antigens that we have become targeted by our own immune system, and therefore can lead to severe consequences. Examples include MS, which I'm sure you've all heard that abbreviation before, short for multiple sclerosis. And I'm giving you just very, very brief descriptions of what happens. But in the case of people with MS, the white matter of the brain and spinal cord are destroyed. And that can be very, very detrimental to their overall health, obviously. Myasthenia gravis impairs communication between nerves and skeletal muscles. So that's obviously not a good thing. We've talked about type 1 diabetes before, which in that case is going to destroy the pancreatic beta cells that produce insulin. And that's why type 1 diabetics become insulin dependent. They require injections of insulin as a result because they're not producing enough or any of it. Rheumatoid arthritis destroys joints. Now, when you think about arthritis, you're probably not going to think about kids. You're going to think about grandma and grandpa, right? Well, that's actually osteoarthritis. That's what happens like as we get old and our joints start to kind of break down. Rheumatoid arthritis is actually much more common in kids and young adults, and it's because it's an autoimmune issue. Graves' disease is antibody-induced stimulation of the thyroid gland. We've actually mentioned that one before. And then lupus is where the body's immune system attacks on tissues and organs, including a whole host of things: the joints, the skin, kidneys, blood cells, brain, heart, and lungs. So that's a pretty severe situation. Therapies and treatment for various autoimmune diseases include removal of the thymus gland, injections of beta interferons, immunosuppressive drugs, and plasmapheresis, which is kind of a cool process where they actually filter their blood and remove antibodies and antigen antibody complexes. And it would have to be done, every so often to be effective. It couldn't just be a one-shot type thing. Another unfortunate side effect of having a active immune system are allergies. And I've promised we would talk about these more, and I've mentioned them a few different times. Allergies happen when a person is overly reactive or hyperreactive to a substance tolerated by most other people. The term allergic or having allergies can be replaced with the term hypersensitive. Okay. An allergen is just the antigens that induce an allergic reaction. So it's anything that triggers an allergic reaction. Common allergens include certain foods, milk, peanuts, shellfish, eggs, various antibiotics like penicillin and tetracycline, venoms that bees, spiders, and snakes might have when they bite you or sting you, cosmetics, poison ivy, pollen dust, and mold. Now, there are two different types of allergies, immediate and delayed. In the case of immediate, we see these symptoms within seconds to minutes. For me, it's like walking into my wife's stepmom's house because she's got cats, and I start sneezing and itching and all that wonderful stuff within a short amount of time. Okay. In the case of immediate hypersensitivity, it's caused by an abnormal B cell response to the allergen. Whereas in delayed hypersensitivity, it's an abnormal T cell response, and the symptoms take a day to 3 days for exposure or after exposure for the symptoms to set in. Okay. Of those things that I listed as allergens, can you think of anything that you might come into contact with, and then a day or 2 or 3 later, you might start experiencing some symptoms of being allergic? Most people are going to experience this. Most people are allergic to poison ivy. Very good. And some people, like my cousin, I know, if we were just out in the woods or something and he sees it, he's pretty well screwed. Some people aren't quite as reactive to different things. Myself, I've never really, I've been in poison ivy before, and I just have a little bit of a rash a couple of days later, but never anything quite as severe as some people have. So I'm pretty lucky, but I'm allergic to a lot of other stuff, unfortunately. Okay, so let's talk about the antibodies that are involved. It's an abnormal IgE class, as we learned, that's involved with allergies. And in response to the first exposure to certain allergens, some people produce IgE antibodies that bind to the receptors on the surface of their mast cells and their basophils. We call this process sensitization, and we can see that taking place right here. Okay, allergen binds to the B cells, which then transform the plasma cells that produce the antibodies which then bind, as you can see, to the mast cells. Okay? And then we see that right there. There's the sensitization. Okay? The next time that same allergen enters the body, it attaches to the IgE antibodies already present. In response, the mast cells and the basophils undergo degranulation, which means that they release histamine from preformed granules. And I've talked about how histamine is annoying, which is why people take antihistamines. After degranulation occurs, mast cells and basophils also release leukotrienes and prostaglandins. Together, those three different chemicals function as inflammatory agents that do things like dilate your blood vessels. So that's why it might appear red. It's vasodilated. They increase blood capillary permeability. Remember, that's where stuff can leave the blood, like the fluid, which can trigger a little bit of swelling. They also trigger contraction of the smooth muscles in the airways, which makes it kind of more difficult to breathe, or you might feel a little bit wheezy. They increase mucus secretion, so that is why your nose might run and you might start sneezing. And they can stimulate nerve endings that cause pain and itching, which people experience, especially like if they've been exposed to poison ivy. So that's what it is. It's basically you've got these specific antibodies that trigger the release of those natural chemicals that we have. And it's just kind of an annoyance that we all experience. All right, last thing I want to talk about is just briefly HIV and AIDS. AIDS is not a word, it's actually an abbreviation for acquired immunodeficiency syndrome, which is a condition in which the person experiences a telltale assortment of infections due to progressive destruction of the immune system cells by the human immunodeficiency virus. Okay. So HIV is also an abbreviation, which is short for the actual virus, which destroys our helper T cells. Okay. Once in our body, it targets those helper T cells, and over 10 billion of those viral copies can be produced each day, and they bud so rapidly from the infected cell's plasma membrane that the cell dies or it lyses, it breaks open, releasing all those viral copies. And so essentially what happens over time is those helper T cells start dying off, and as a result, our acquired immunity starts to be deficient. Okay? And that's why they call it immunodeficiency syndrome. Your immune system is not working to its highest capacity, which then can lead you to get sick more easily, to what we call opportunistic infections. These are diseases caused by microorganisms that are normally held in check, but now can proliferate because the immune system is defective. So in other words, when you have this AIDS progression, it's because your immune system is so compromised that you're going to get in big trouble just from something simple that, like, the flu or some other common illness a normal person would be able to fight off. But since your immune system is so compromised, you're at a disadvantage and could lead to death. Now, there's been a lot of remarkable treatments and HIV and AIDS that have attenuated it quite a bit, but it is still an unfortunate situation. They haven't completely cured it. Anyway, that's the immune system. Pretty darn complicated, but very amazing, and obviously at the forefront of everything we think about right now, because we're in the middle of a pandemic. So with that said, we are officially done with unit, whatever this is 5, I think. And we're ready now to move on to unit 6. Unit 6 is one of the most complicated systems, or units, I should say, that we will talk about this semester. My screen will pull up. We will be covering 2 systems in this unit. The first of which is the respiratory system. The second of which is the digestive system, which actually relate to one another quite a bit. You might not think about that, maybe you do, but breathing and eating are processes where we bring something into the body that our body doesn't naturally produce. In the case of the respiratory system, we're breathing in oxygen. in the case of the digestive system, we're ingesting, water, electrolytes, carbohydrates, proteins, so on and so on, so on. Okay, so we're needing to bring stuff in, and those are actually the ingredients that we need to make energy. Okay, but as a byproduct of both breathing and making energy and digesting our food, we also produce waste products that we must get out. In the case of the respiratory system, we're talking about carbon dioxide, which we've learned very recently. If you don't exhale your carbon dioxide, it can lead to acidic environments inside our body, because what it will do with water is form an acid. Whereas the digestive system, we're talking about make a poop, and we'll get into that obviously here in the next couple of class periods. So there's three major things that we can say our respiratory does to contribute to homeostasis. Number one, I would say, is the primary function, which is providing for exchange of gases, oxygen, carbon dioxide between our atmospheric air, but also our blood and our tissue cells. By exhaling that CO2, I just mentioned, that helps regulate our blood pH. And then kind of secondary functions there are containing receptors for the sense of smell, filtering inspired air, producing sounds for, talking vocal sounds, and excreting small amounts of water and heat. So we're actually going to talk a little bit about acid-base balance, regulating pH in this lecture, just very briefly. We introduced at the beginning of the semester when we talked about acids and bases and pH scale. And I said that there are two main systems that are most responsible for regulating pH. One of them is the respiratory system. The other, we have yet to talk about it. It's not the digestive. It's the urinary. And that's what we'll get into in our last unit. Let's just start here by introducing the structures and what we would call the divisions of the respiratory system. Kind of like what we usually do. A little background on the structures and their anatomical features so that we can understand how they work. There's kind of two major ways to break up the different structures of the respiratory system. First way is to divide it structurally. The second way is to divide it functionally. Okay. If we look at the structural classification, there's two different tracts or systems, what we call the upper and the lower. The upper respiratory tract or system includes the nose, the nasal cavity, which is the inside of our nose, and then the divisions of our pharynx, which is a fancy term for our throat. Then you have the lower respiratory tract or system, which includes the larynx, which we commonly call our voice box, the trachea, which we commonly call our windpipe, and then the bronchial tree, which is a series of airways on the inside of our lungs, and then the lungs as a whole as well. Then the two divisions of the respiratory system based on their functionality include the conducting and the respiratory zones. The conducting zone is going to filter, warm, moisten, and conduct air into the lungs. Okay, hence the name conducting zone. It conducts, it moves the air into the lungs. It goes from our nose all the way down to what we call our terminal bronchioles. Then the respiratory system or zone, consists of tubes and tissues within lungs where gas exchange actually occurs. So this is where oxygen actually enters the blood. CO2 is able to leave the blood to then be exhaled out. It includes respiratory bronchioles, alveolar ducts, alveolar sacs, but more importantly than any are these structures called alveoli. And we're going to learn more about them later on in the lecture. But those are these like tiny, and I'm talking microscopic sized balloon-like structures that are surrounded by our pulmonary capillaries, where the true exchange of the gases takes place. Now, those are all structures located inside your lungs, and your lungs are located in the thoracic cavity, which also consists of the heart, large blood vessels, trachea, esophagus, and thymus. Not that that's really important, but I listed it off anyway. Surrounding our lungs are two wet epithelial membranes, and they are a parietal and a visceral pleura. Okay, and these are very important because they affect breathing in and breathing out. Okay, they are pretty similar to the pericardia that we talked about the other day that surround our heart. Okay, surrounding your heart, we learned that there's a parietal and visceral pericardium. In the case of the lungs, these are now the parietal and visceral pleura. Parietal pleura is a thin superficial membrane, okay, which lines the thoracic cavity, and then the visceral pleura, which covers the lungs. Okay. In between them is a small, and I'm just going to describe it like a potential space. We call this the intrapleural space. Okay. It is full of a fluid called pleural fluid that reduces friction between those two membranes, but also produces surface tension that causes the two membranes to stick together. Okay. And this is important because this is, as like I said, going to affect your ability to breathe in and out. Okay. Within this space is essentially a vacuum, not like the type of vacuum that you use to clean your floors, but it's a established or maintained pressure. Okay. And that's what helps keep those two membranes along with that fluid sticking to one another. I always think about like their relationship, like from a movie that I watched as a kid, Little Giants, where there's a scene where a boy, he's trying to play football, can't catch anything, so they put some glue stuff on his hands, and they send him out and they're going to throw him a pass, but right before he puts his hands together and they get stuck, and then he pulls his hands apart and they snap back together. And that's what these two membranes like to do to one another. They like to be attached. Okay, so if you pull the parietal pleura in one direction, the visceral pleura is going to go with it. And you've probably heard of somebody having a collapsed lung, right? A collapsed lung would occur when those two membranes are no longer sticking to one another. There's no longer that vacuum in the intrapleural space. Okay. Now, I'm kind of getting ahead of myself, but that's one of the things involved with breathing, which is one of the 5 processes of respiration. Respiration is a fancy term for the process of gas exchange in the body, which begins with breathing, what we would call pulmonary ventilation, if we were going to be a little more specific, where air flows into and out of the lungs. Movement of air into the lungs is called inspiration or inhalation, whereas movement of air out of the lungs is referred to as expiration or exhalation. And when we inspire, this allows O2 to enter the lungs, and when we expire, this permits CO2 to leave the lungs. Okay. That's obviously what's shown here at the top with our figure, okay, where we're bringing in O2 and we're pushing out CO2. We'll talk about that here in just a second. Okay. Now, in this illustration, we see that term alveoli that I mentioned just a second ago. And I told you guys that's where the gas exchange occurs, where actually oxygen is able to enter the blood and CO2 is able to leave the blood. Okay. If I go back to this slide, listed down there as part of the respiratory zone are what we call alveolar ducts and alveolar sacs, along with alveoli. And I'm just going to kind of skip over respiratory bronchioles. Okay. What we see here is a cluster of alveoli, which is what an alveolar sac is. I always tell students, think of alveoli like grapes. Okay. Each individual ball-like structure is an alveolus, which is the singular form of the word alveoli. Okay. And like grapes, they don't grow in solitary situations. They grow in clusters. So a cluster of alveoli is an alveolar sac. Okay. And like the stem that holds those grapes together, that's what this is, and that's what the alveolar duct is. That's an airway that leads to all those little sac-like grapes. And that's where, and what's not shown, but pulmonary capillaries will be very prevalent surrounding each one of those for ready gas exchange. The type of gas exchange that occurs in our lungs is called pulmonary gas exchange. Also goes by the term external or pulmonary respiration. And this is where we exchange the gases between the alveoli or air sacs of our lungs and the blood in our pulmonary capillaries. In this step, pulmonary capillary blood gains O2 and loses CO2. Okay. The third component of respiration is the transport of oxygen and carbon dioxide by our blood, which relates to what we've been talking about here for the last couple of class periods, which, you know, thinking back to the day, we talked about the blood and things like hemoglobin. We're going to talk more about that here again in this lecture. We'll get to that point today, though. Okay. So this is where we are going to be transporting oxygen to our systemic tissue cells. So from like the top of our head down to the bottom of our feet, where we're going to carry that CO2 the opposite direction to the lungs to be exhaled out. Once we get that oxygen to the tissues, we need to bring that oxygen into the tissues, which is what we do with step 4, systemic gas exchange, which goes by also internal or tissue respiration. And in doing this, the blood loses the oxygen because it goes into the cells. CO2 is removed from those cells and is pulled into the capillaries to then eventually be exhaled out. What we're not going to get into in this lecture is number 5, which is cellular respiration, where we use that oxygen, we produce CO2, we produce a little bit of water, but more importantly, and the whole reason that we do any of this is to produce ATP, produce energy. And this is going to involve processes like glycolysis that we've mentioned before, Krebs cycle, the electron transport chain, which if you've taken biology or any other form of science class, I'm sure you've been exposed to it, whether it was freshman year of high school or in another college class. That's actually going to be what we're going to talk about in the last lecture of this unit. And that's our cell metabolism lecture. And that will be what we discuss after we've talked about the respiratory and the digestive systems. Because the respiratory and the digestive systems provide us with the ingredients, the nutrients like glucose from carbohydrate loaded foods, oxygen, which we get from breathing. So that's why we talk about those two systems and then talk about the wonderful processes of cell respiration. Okay, so let's talk pulmonary ventilation. That's our very first of the 5 steps of respiration. In other words, that's what we would commonly call our breathing processes. Consists of a repetitive cycle of inspiration and expiration. We call this constant alteration between inspiration and expiration, the respiratory cycle. We, on average at rest, have about 12 breaths in, 12 breaths out per minute. Okay, that number can go way up or it can slow down depending on the state of the body. Just kind of like what we learned with heart rate. When we exercise, your heart rate goes up, so does your respiratory rate. Okay. Now, how this all works is very similar to what we learned about when we talked about the flow of blood. And as I promised, when we talked about that, we would talk about this again. We're talking about pressure gradients. Okay. And in the case of breathing, we have several laws that we're going to see in this lecture, but the one associated with breathing is what we call Boyle's law, which states that there's an inverse relationship between the volume and the pressure inside of a closed container, i.e. our lungs and the thoracic cavity. Okay. What that means is if we have an inverse relationship, one goes up, the other one goes down. Or if one goes down, the other one goes up, right? We've seen that several times here lately, like in the case of vascular resistance and blood flow, that was an inverse relationship. So here we have kind of a easy experiment to demonstrate this relationship. We've got a closed container, okay, full of air with a piston that can change the volume inside. So in other words, can change the amount of space on the inside of the container. Okay, with the piston in the up position, we say that the volume would be measured out at 1 liter, and the pressure at that point is 1 atm. That's short for atmosphere. Okay. Well, here the volume is pretty high and the pressure is pretty low. Whereas if that piston pushes downward, the volume goes down to a 1/2 liter, and in essence, that drives the pressure up because there's more stuff, or there's the same amount of stuff in a smaller space. The same thing can happen like if you think about like a juice box, okay, or a water balloon. A water balloon is a good example. Okay. If you squeeze a water balloon, what happens to the pressure inside? It goes up, right? Because there's less space for that same amount of water. Okay. Well, how does this relate to breathing? Well, we can change the dimension of our thoracic cavity where our lungs are either increasing it or by decreasing it, which is going to affect its pressure inside. And this is what we do to inspire and expire. Okay. Inspiration is the movement of air into the lungs, occurs when atmospheric pressure, what we would say is at sea level 760 mmHg, there's that millimeters of mercury, or we abbreviate that P sub B, which is short for barometric pressure. So that's the pressure in the atmosphere that we're breathing. If that's greater than alveolar pressure, so in other words, if P sub B is higher than the pressure in our lungs, our alveoli, P sub A, we've got a pressure gradient that's going to draw the air into the lungs. Now, how do we drop pressure in the lungs? We make our closed container larger. You'll see on the next couple slides how this all works. When we expire, we move air out of the lungs, and this occurs when the pressure inside our lungs in the alveolar spaces is now greater than the atmosphere. So P sub A has to be greater than P sub B to push the air out. So how do we change those pressures? And we have to change the dimensions. And we have to contract mainly our diaphragm as well as some other muscles, or relax it in those muscles. And we'll see here in just a second. So to kind of emphasize how it all works, alveolar lung pressure changes as a result of changes in intrapleural pressure. Remember, intrapleural pressure, P sub IP, would be the pressure in the space between those two membranes around our lungs. Okay. During the respiratory cycle, so in other words, as we breathe in and out, several mechanisms are used to change the size of the thoracic cavity, which in turn changes intrapleural pressure. So if our thoracic cavity dimensions go up, that means the space has gone up. What would happen to the pressure in the intrapleural space? We said it's an inverse relationship between pressure and volume, right? So if our volume goes up, pressure here goes down, right? Okay, if pressure in the intrapleural space goes down, so does pressure in the alveoli. Alveolar pressure drops as well. That establishes a pressure gradient that's going to result in air coming in. What if our thoracic cavity dimensions decrease? So volume goes down. What would happen to pressure here now? It would go up, right? Because it's obvious, or it's an inverse relationship. One goes down, the other one goes up. So dimensions go down. That means our volume's gone down, pressure goes up. Pressure also in the alveoli go up. Now the pressure gradient is established, that forces the air out. We exhale. So how can we change the thoracic cavity dimensions? Well, in the case of inhalation, I've already mentioned, the most important muscle is your diaphragm. Other term for inhalation is inspiration, and that's just the process of breathing in. And for this to occur, the air pressure in the lungs must decrease below pressure in the atmosphere. And this is accomplished by expanding the lungs to increase their volume. Volume goes up, pressure goes down. Most important muscle, as mentioned, is the diaphragm. This is a dome-shaped muscle that forms the floor of the thoracic cavity. And by contracting it, we can say that this is an active process, because what we've learned previously, contracting muscles is pretty complicated. This is the skeletal muscle, so it's got the actin-myosin, it's got troponin and tropomyosin, all that wonderful stuff, and it's very active. Okay, major muscle we can describe as being used when we quietly inspire. What we're all doing right now is quiet inspiration, which is just where you're taking normal breaths in and out. A quiet normal breath in is quiet inspiration. A quiet normal breath out is quiet expiration. Okay. Now the next most important muscles of quiet inspiration are your external intercostal muscles. These have the ability to pull the anterior end of each rib up and out, enlarging the thoracic cavity from front to back and side to side. Okay. Now, if everybody takes a deep breath in, take a deep breath in. you just performed what we call forced inspiration, which involves other skeletal muscles, namely the scalenes, your pectoralis minor, and your sternocleidomastoid. These are located kind of in the upper chest and even into the neck, which is where your sternocleidomastoid is. And they help increase that thoracic cavity size even more than it already was. Okay. So when the diaphragm contracts, it goes from a kind of dome shape like we show here to more of a flattened shape. Okay. And in doing so, that pulls on those two membranes we just talked about. Okay. And remember, those membranes stick to one another like a kid in the movie Little Giants with his hands glued together. If the parietal pleura is pulled down with the diaphragm, so is the visceral pleura. And in doing so, that pulls the lungs with them. And so the volume goes up and the pressure goes down. Look what happened to alveolar pressure and intrapleural pressure here versus now here. Doesn't have to be a drastic change in pressure, but it is enough that now pressure in the lungs is less than pressure outside the lungs. 760 is more than 758, correct? So that's a pressure gradient that's going to draw the air from high pressure to low pressure. So I've already kind of explained it, but let me read through. As the thoracic cavity enlarges, it pulls the lungs along with it because of the cohesion between the parietal and pleural membranes. Thus, the lungs expand and the pressure in the bronchial tree decrease. The ability of your lungs and the thorax to stretch is called compliance. Compliance in the lungs is related to two principal factors: elasticity and surface tension. What you want is your lungs to have high compliance and thus can expand very easily because the elastic fibers in our lung tissue are easily stretched. And then there's also what we call surfactant, which we'll learn a little bit more about, in our alveolar fluid that reduces surface tension. Okay. Think of a rubber band. And we've talked about this before. A rubber band has the ability to stretch, but then also to recoil, right? Well, if you think about like going to Walmart and buying a bag of rubber bands, you can buy like the variety bag of rubber bands, where you've got some that are really thick and really strong, or some that are really thin and much easier to stretch. Okay? Or you think of like if you work out and you use like those TheraBands that have different colors, some are easier to stretch than others. Okay. What you don't want is your lungs and your membranes of the thorax to be like a really thick rubber band. What you want is that thin rubber band that's easy to stretch. Okay. But there are conditions, unfortunately, that can decrease compliance and thus make your lungs more like a really thick rubber band, or what is it, like the black TheraBand? That's the really hard one to stretch. Okay. Pulmonary conditions that decrease compliance include scar lung tissue, where the lungs are obviously scarred. Lung tissue becomes filled with pulmonary fluid or fluid in the lungs, pulmonary edema. There's a deficiency in surfactant, or where there's an impeded expansion of the lungs in any way, like in paralysis of the intercostal muscles. Okay. So that's decreased compliance. Okay. And the people that have decreased lung compliance, their lungs don't stretch, so they can't take like a really deep breath in. Now, what you also don't want is too much compliance. Okay. What you don't want is like what can happen to your favorite pair of sweats after a couple of years of wearing them all the time, where the waistband gets too stretched out, that it no longer likes to snap back, right? It's easy to pull and stretch them out, but now they don't snap back. So there's conditions that can increase lung compliance. And this includes emphysema, which can be due to destruction of elastic fibers in their alveolar walls. Okay. And so people with emphysema don't have any problem taking a deep breath in. What's the issue with people with emphysema is blowing that air back out. And that's what exhalation is. So in the case of exhalation, it occurs when the pressure gradient resulting in inspiration are now reversed. So in other words, pressure now has to be higher in the lungs than it is in the atmosphere 762 is higher than 760, so that forces the air out into the atmosphere. Okay. Now, unlike quiet inspiration, when you're quietly breathing out, we call this a passive process because it only involves relaxing the muscles that we just previously learned contract when we're normally inspiring quietly. So in other words, when we quietly exhale, it's basically just a passive process because it's involving the diaphragm just relaxing, going back to its original dome shape, which in doing so pushes the lungs to decrease their size, decrease volume, increases pressure, and pushes the air out. Does that make sense? So that's why we said quiet inspiration is a active process, but quiet expiration is passive. Now, what this does require, though, is a dependency on your lungs to return to their initial size, causing a decrease in the size of the thoracic cavity. And we call this ability of our lungs to return to the initial size elasticity or elastic recoil. Okay. And that's, like I said, what people that have a stretched out waistband in their pants don't have. Okay. Individuals with emphysema are like those stretched out waistbands, where they stretch really easily, but the recoil is gone. One of the telltale signs of somebody with emphysema, if you've never been around them, aside from probably having a gas mask hooked up to their nose all the time, is that their lungs, they describe it as being barrel chested. Basically, their lungs are just like overinflated all the time. They can't just puff and blow everything out. They can't even exhale completely because their lungs are overstretched. They don't snap back. They don't recoil. Okay. Now, speaking of huffing and puffing and blowing out, that's what forced expiration is. Okay. Taking a deep breath in, forced inspiration, blowing that air back out is forced expiration. That involves recruitment of the muscles of exhalation. Okay. That includes your internal intercostal muscles. So your externals help with breathing in, internals help breathing out, as well as your abdominal muscles. And they can increase pressure tremendously and establish a pressure gradient up to 20 millimeters of mercury that forces the air out pretty darn quick and pretty significantly. Does that all make sense? Now think about emphysema. Pulmonary edema can be caused by certain conditions. Lung tissue scarring. What is a common theme in a lot of those situations? In other words, what do people do that causes them to end up with emphysema? Smoke. And now vape. Okay. A lot of lung issues are self-induced. People know that. Unfortunately, they choose not to pay attention and do it anyway. So it's unfortunate. Now, like the flow of blood through blood vessels, the rate of airflow through airways depends on both pressure gradients and resistance. So we talked a lot about resistance in our blood vessels lecture. Systemic vascular resistance, we learned, is inversely related to the flow of blood. Well, we have airway resistance, kind of the same concept, except it's not blood that's flowing, it's air. And resistance affects both inspiration and expiration. Okay. What we want is very little resistance. Okay. And the walls of our airways, especially our bronchioles, offer some resistance to the normal flow of air into the lungs. Okay. The larger the diameter of the airway, the less resistance they have, which makes sense. They're more wide open. There's less resistance to the flow of air. Okay. As our lungs expand during inspiration, the bronchioles enlarge because their walls are pulled outward in all directions, which decreases airway resistance. And that is what happens when the diameter of the bronchioles decreases. Okay. Airway diameter is also regulated by the degree of contraction or relaxation of smooth muscles in the walls of the airways. So we talked about vasoconstriction, vasodilation, and how those can affect resistance. Now we have bronchoconstriction and bronchodilation. Okay. Bronchoconstriction, bronchodilation is essentially the same thing, except now we're not pinching or opening up blood vessels. It's airways. Okay. Bronchodilation decreases resistance because our airways are more open And this is what we do when the sympathetic nervous system sends out its signals, which should make sense because as we know, if we're in a fight or flight response, we're not only breathing more often, we're going to be breathing more deeply. So we want that resistance to the flow of air in and out of the lungs to be diminished, right? Whereas when the parasympathetic division is sending out its signals, we're going to trigger bronchoconstriction, which increases resistance and less air is able to flow through. But that's okay because we're in a rest and digest response. We're not needing to move tons and tons of air. Now, there are conditions that can narrow or obstruct the airways, which increases resistance so that more pressure is required to maintain the same airflow. It's kind of like what we talked about with atherosclerosis. A hallmark of asthma or chronic obstructive pulmonary disease, COPD, which includes both emphysema and bronchitis, is an increased airway resistance due to obstruction or collapse of their airways. We'll talk a little bit more about those in a little bit. Probably not today, though. Actually, you know what? I'm going to stop right here because I don't want to start this. We're not going to have enough time to get through like the next 4 slides where we talk about all this. So we'll stop right here, and then we'll start fresh with pulmonary volumes Tuesday after we get through the exam, and then run from there. We got a pretty good jump, so I'm happy with where we're at.